Thank you, everyone, for showing up on a beautiful day. The weather's been so terrible on the water, it's actually kind of frustrating to be here tonight. A neighbor of mine just told me that the bait are jumping on the beach in front of the house, and here we are. So thanks for coming. Bonefish. Why should we care about bonefish? And I'll get to a lot of reasons why. But from a purely scientific reason, they're extremely challenging to understand. And there's nothing better that you can do to a scientist than to challenge him or her to get their blood going. So I'm going to start out with some background on bonefish and then move into some of the research that's ongoing in the field as well as in the lab. Okay, the genus Albula, there's a number of species. It's been around in the present form for 138 million years. There aren't a whole lot of fish swimming out there that have been around that long. The challenge is, despite them being in shallow tropical seas, so easily accessible to people, they're not like deep sea creatures. There's a lot of species worldwide, but it's really tough to tell them apart because their characteristics are what we call highly conserved. So even though these species have been evolving over the past 138 million years from just 2 that long ago to 12 now, you could have all 12 species in that photo right there of the bonefish school, and you won't be able to tell which species that you have. We've got to do genetics on it. Typically, we'll use different morphometrics, say like the number of spines in the rays, the fin rays, to differentiate species of fish. So one species might have 6 rays in the dorsal fin, another might have 15. The overlap for all these different bonefish species is so extreme, say 6 to 8 in one species, 7 to 9 in another, that you really can't differentiate them. So it wasn't until genetics got much better than it was, say, 20 years ago, that we were able to start looking at species. So figuring that out, especially since a lot of these species occur together, you can actually catch 2 of the same species, for example, on the edge of a flat. It's made it very much a challenge to figure out what's going on. So the distribution of bonefish around the world is shown in this graph here. You can see tropical and subtropical shallows globally, pretty much, all have bonefish species. In Florida, we're limited to the Florida Keys for the most part, up through Biscayne Bay. This graphic shows the species that have been ranked as species of concern, say vulnerable, threatened, in one case endangered, by the International Union for the Conservation of Nature. A few years ago, we did a species assessment and classification. And that brings us to the species that we're focused on here in Florida, Albula vulpes, which is in the Caribbean. That's a picture of an Albula vulpes from the Bahamas. They've been around in this form, this species, for 11 million years. And we can tell that from basically the genetic clock, right? So mutations occur at a pretty much standard rate in our genes. And so we know from fish, from that genetic clock, that they've been around in their present form as a species for about 11 million years. They're limited geographically to just the Caribbean, the Florida Keys, and the Bahamas. Compared to other species of bonefish around the world, Albula vulpes, the one in this region, has the smallest geographic range. Most of the others have much broader geographic range, which is kind of interesting given some of the stuff I'll show you in a bit about their connectivity to other populations. One of the reasons that I study them and others study them is because they're economically valuable fisheries, or they support economically valuable fisheries. There's a very valuable recreational fishery in Florida, Bahamas, Belize, Cuba, other places. But there's also a lot of subsistence fishing for bonefish, as well as some commercial fisheries. Cuba, for example, has a pretty intensive commercial fishery. So they're accessible, they're close to shore, they're economically valuable, and if you follow any kind of fisheries management around the world, that typically means that they get to be in overfished states, and in some places for bonefish, that's the case. The real challenge is we have limited biological knowledge. I could not have stood up here 15 years ago and told you much about what I'm going to tell you here over the next few minutes. We just didn't know much at all. It would have been maybe 3 slides and that was it. But as you'll see, I've got a lot of information to share, but that's all very recent. So let me do back up on that one. That IUCN classification did classify Albula vulpes as near threatened. That means that it's at risk for becoming what we would call vulnerable, which in fisheries terms you would consider overfished and something in need of attention. But the IUCN assessment doesn't just look at overfish status or fishing harvest. It also looks at the effects of habitat loss, degradation, and a number of other factors for classification. And I'll touch on all of those here in the next few minutes. So I'm going to go through the status of knowledge of bonefish, what we know about them from the wild, a lot of research, and then turn it into a conversation about some of the lab work that we're doing here at Harbor Branch. So the first thing we did, starting about 15 years ago with bonefish, is try to figure out some very basic things. What habitats did they use? How far did they move? How long did they live? Super basic things. So number one was figure out what we call their home range. Were they the kind of fish that would not go very far, live in a very small area, or they kind of wandered around? You might think of bluefin tuna as a type of species that wander around a lot. Some of the coral reef fish that folks here work on, like damselfish, they don't go anywhere. They're very site attached. Where do they spawn? Until 2011, we had no idea where bonefish spawned. And do they migrate to spawn, or do they spawn at the same place where they live throughout the year? All basic questions. So the way we started to address that was with dart tagging. You inject these tags into the dorsal musculature of the fish. They have identification codes on them, numbers, and they also have the website for Bonefish Tarpon Trust as well as a phone number. So if people catch these, they can call it in, let us know where and when they caught it, and we can start to figure out movements. So we work a lot with fishermen. They did a lot of the tagging, as you can see from this picture. Fly fishing is a pretty popular activity for bonefish. But we would also collect or catch bonefish in large schools using, say, 600-foot seine nets. We could sometimes tag 500 or 600 in a day. So as to give you an idea of how many fish have been tagged, in the Florida Keys, 7,000 fish were tagged. That project ended about 8 years ago. In the Bahamas, we're still tagging. I think we're up to about 13,000 fish tagged there. Belize in Mexico, I think we're up to about 6,000. The reason you have to tag so many fish is your recapture rate is typically pretty low. In a tag recapture study, if you get 5% of your tagged fish recaptured, that's a pretty good number. In bonefish, we're between 3 and 5%, depending on where we are. If you get a lot higher recapture rate from that, that probably means that you're catching nearly all the population, and you've got to probably have an issue there too. This is what a tag looks like after it's been on a fish for about a year. This particular fish was tagged and recaptured in Cuba. It was recaptured exactly 365 days after it was tagged in exactly the same place it was first caught, a place about half the size of this room, which is pretty insane. And I'll get more to that in a bit. So basically what we found is that about 75% of the fish that are recaptured within a kilometer where they were tagged. And just very quickly, I'm going to show you some of the places. This is Abaco in the Bahamas. We've been working for quite some time. Each of the colored dots, similar colored dots, represent the tag and recapture location for a single fish. And you can see they're all very close to one another. Almost all the fish that are not caught close to their tagging location, that had to do with spawning migrations. So we've been able to identify what we would call this a home range for this population of adult bonefish. To get at the question of where do they spawn, do they migrate to spawn, we switched to a different approach, and these are acoustic tags. You can see there in the center, that's an acoustic tag, about this big. We surgically implant them into the bonefish. This is a bonefish that's upside down. We slice the abdomen, clear the scales, slice the abdomen, insert the tag, and then use sutures to close it. These tags, you may have heard a lot about radio tagging, say birds or freshwater fish. Essentially, a radio signal is an electronic signal. Electronic signals in salt water are attenuated or disappear very, very quickly. So radio technology in salt water doesn't work very well. So instead, these acoustic tags, they put out a subsonic ping. The way to think about this is, say, your favorite submarine movie. Those are basically acoustic, subsonic pings that they're putting out. The machines allow the people in the subs, or in our case, the computers, to translate those subsonic pings into levels that we can hear. On the left side there, that's an acoustic receiver. We mount those underwater. We anchor those underwater. If the fish swims within, oh, say 500 meters or 500 yards of that receiver, its tag gets picked up. Each tag has a unique ping. We can identify each individual fish with that code. On the upper right there, you have a computer that we can use to actually monitor fish in real time. If we can find a fish and follow them around with a hydrophone we have over the side of the boat, we can actually follow fish in real time and get that type of information. When we can do that, we can get information off these tags if they're the correct type of tags of water temperature, the direction that the fish is moving, whether up or down or sideways, how fast it's moving, as well as pressure, so we know its depth. And that'll be important here in a moment. So with those acoustic tags, the yellow is the home range that I just showed you. Working with fishing guides who started to tell us about some of the things they were seeing off the flats, we started to home in on a spawning site. So we started to get fish moving long distances by putting acoustic receivers in different places, such as the point there next to, right here next to that red circle. Any kind of constriction point that was good for an acoustic receiver. Up here on this point was another, and we could kind of track their migrations. Another one, a little bit shorter migration. That longer migration round trip is about 140 miles. And if you remember, these fish are only maybe 23, 24 inches long, so they're going a long ways. And then we also get them coming back. Remember I said with home range, they come back? Now think about this. A lot of us go into a shopping mall, we can't come out and find our cars. Right? These 25-inch fish, 23-inch fish are able to go 140 miles round trip, a week later come back and go to the exact same spot. It's pretty incredible. Trying to figure out how they do that, that's someone else's challenge, not mine. So what they do is they go to places, you can see here, shallow flats as home range. They can get to the spawning area by going along a shallow shoreline. But when they get to the spawning location, that's quite a drop-off. So what happens is they gather in shallow bays near deep water. This is a school of bonefish right here, probably about 4,000 or 5,000 bonefish in the shallow bay getting ready to spawn. When they spawn, we've got a video here that I'll show you exactly what happens. Late in the afternoon, about 3 o'clock, they gather in large schools. And this is a school of about 10,000 fish in about 40 feet of water. You can see some behaviors here. Up at the top, you can see bonefish coming off the school. They're leaping out of the water. And what they're doing is they're gulping air. When you swim with these fish, you can actually see bubbles coming out both ends. They're farting air and they're burping air. What we think is happening is they're filling up their abdomens with air. The eggs are still pretty small. Then they migrate offshore. When they migrate offshore, they would do a deep dive. You can see on this slide at about 5 o'clock, which is at dusk, this is winter, they do a deep dive. These are in meters, so multiply times 3 on the vertical axis there for feet. And they dive deep. In this instance, the tags that recorded depth were only good to 50 meters. And they went deeper than that. They kept going. They stay down there for a bit. What we think happens is during that point, the eggs hydrate, take on water, become ready to, and go under other transformations, ready for spawning, and that's when they rush to the surface, or towards the surface, which is this vertical rush right here, up to about 80 feet. We think that's when the spawning happens, when the air expands, forces all the eggs and the sperm out, and that's where fertilization occurs. These fish and most marine fish don't have nests like a largemouth bass or salmon. It's external fertilization that's called broadcast spawning. And then that's it for parenthood. These fish come back in that night. The next morning they're gone. They head home. So it's a long trip, a day to pre-spawn, a night to spawn, and then that's it. They're done. They go home. So over time, each of these yellow pins is a spawning spot that we've identified. So since 2011, we've been able to identify a lot of different spots throughout the Caribbean. When they spawn, the eggs hatch. This is what a larval bonefish looks like. And that's my hand for scale. This larvae is dead or dying, and so that's why it's becoming opaque. When they're alive, they're completely clear, as in you could be swimming right there and you wouldn't be able to see them, which is great camouflage in the open water. They drift around in the open water as plankton for between 40 and 70 days. So a long time that they're out there. The average is about 50. Then they come in close to shore, if they're lucky, and metamorphose into baby bonefish, like this guy here. When they're this small, I'm not sure you can see from back there, but their skulls are still clear. So when you look down, you can actually see their brains in there, which is kind of a strange thing to do. When those larvae come in, it's very specific of what they're looking for. Open ocean here, they need channels that come in, and then these juvenile bonefish, the larvae and juvenile bonefish, are looking for these kind of muddy, open sand flats where they start their juvenile lives. So from a conservation perspective, this is really important because bonefish use the entire coastal habitat mosaic. Number one, the home range, which I just showed you. And on this slide, right here is a great place to go fishing for bonefish to live. We know that they undergo spawning migrations, right? So they're using a lot of the coast. From here, they'll migrate all the way out to this point here to spawn. They collect in pre-spawning sites in those bays. Then they go offshore to spawn, right? So we're already talking about 4 or 5 different habitats. Then the larval transport, right, which could be local or could be long distance. I'll talk about in a second. The juvenile habitats are very specific, back inshore, shallow, muddy bays, and they're very economically important as a fishery. From a conservation perspective, that qualifies them as what we call an umbrella species. They're charismatic. They use a lot of habitats. If you protect bonefish, you protect a lot of other species in the coastal zone, which are not going to be nearly as sexy to protect. Right? Think about bonefish versus something like a killifish or mosquito fish. A lot better chance of protecting bonefish. So the work that we're doing both in the field and here is not only specific to bonefish, but it helps at a system level as well. We've also been working a lot on population connectivity. Mitch Roffer, who's here, worked with us on some computer modeling to say, okay, if bonefish spawned in location X, in this case down in Trinidad, where would those larvae be transported, knowing what we do about the ocean currents and actually using real ocean current data. So this slide shows where all those larvae could be going over the average of 53 days. We'd run the model for 53 days. Bonefish spawn at new moon, or full moon, excuse me, sometimes new. We'd start that at the full moon in, say, October and run it. Many different simulations. You can see a lot of potential connectivity here just from Trinidad because of the currents. Next spot, on the south coast of Cuba, a place that we've identified as a spawning location. You can see a lot of connectivity to the Yucatan. Then they get caught in the loop current and Gulf Stream and show some connectivity to South Florida as well. If they spawn at a place that we're investigating right now, west of Key West, there's a lot of what we call self-recruitment. Because of the currents, a lot of those larvae are retained there and come right back into the Florida Keys. So if you're a fisheries manager, it's really important to know where your fish are coming from. If your fish are coming off from somewhere else down in the Caribbean, you need to manage your fisheries, but you also got to talk to the folks who are upstream. And we're starting conversations now with Mexico, Belize, and Cuba because a lot of our fish are coming from upstream, it seems. But we also then have to protect our local habitats, which I'll kind of go into in a minute. We're also in the midst of a genetic study, sampling fish from around the Caribbean to see the level that the different populations are genetically connected. And that's going to tell us a lot about regional management and conservation also. Next, we went on growth, looked at growth. Again, nobody knew how fast bonefish grew. The way we age bonefish, like most fish, is we have to kill them, unfortunately, take out their ear bone, or otolith, and it has growth rings in it, just like a tree. We take a special two-bladed saw, and we can slice a thin slice out of it, and it looks just like a tree ring. And so you can see this guy was at least 8, 9, 10 years old when he was harvested. So if we can collect, say, 100 of those fish from different sizes from a bunch of different locations, we can make a growth curve, and we know on average a fish of a certain length is going to be a certain age. But what we also found is that fish grew at different rates depending on where we found them. So this shows you graphically the 8-year-old bonefish in the Florida Keys versus in the Caribbean islands. Much faster growth rate in the Keys. Another way to look at it, the same size fish. This is a 23-inch fish. In the Florida Keys, it's 6 or 7. You catch this fish, say, in the Virgin Islands or the Bahamas, it's 16. So much slower growth rate. So if you're a manager in the Bahamas and you want to have a good bonefish fishery, you don't want to try and emulate the Florida Keys because you're never going to get there. The fish are just as old. They're just not as large. But that also means if you go to the Bahamas and you catch a 12-pound bonefish, there's a good chance that fish is 30 years old. I know, older than some of the people in this room who probably fish, which is pretty insane. Next, we figured out about diet, right? Because diet has a lot to do with condition, their ability to grow fast, avoid predators, that type of thing. So what do bonefish eat? And it depends. Does anybody know what that is? It's a bonefish tongue. They don't have teeth per se. They have a bony tongue and the top of their mouth is bony and they crush their prey. So if you fish for bonefish and you bring back your hook and it's been bent, the fish ate it, crushed it, and spit it out. And then you feel very bad for missing the fish. So a lot depends on habitat and location. So I'll quickly run through in the Florida Keys, swimming crabs, snapping shrimp, mud crabs, toadfish, worms, all things associated with seagrass beds. And if you've been to the Florida Keys, Biscayne Bay, there's been a lot of seagrass beds, although we're losing them due to pollution. At Turnip Island, Belize, they also have seagrass beds, but they're not the kind that we have in the Keys. They're associated with the back reef, the back side of the coral reef. So it's a lot of rubble, shorter seagrass, completely different prey base for the bonefish. So maybe that has something to do with the growth rate. We're not quite sure. You go to Grand Bahama Island, the Bahamas, there's a lot of sand. There's not much seagrass. There's some algae. And the algae can support similar stuff to seagrass. So you see a mix of the diet there between open bottom and the algae bottom. Go to Andros, Bahamas, where the bonefish flats are entirely sand and mud, and it's a completely different diet. But you do notice on the bottom, fish, they do eat a lot more larger fish over there in Andros, Bahamas for some reason. And they also change their diet with growth. Less than 16 inches, a lot of clams and worms, which are associated with the open bottoms, where those small bonefish hang out. When they get larger and they can avoid predators, much more diverse diet, which is great, so that they can actually build up enough resources, fat, whatnot, to be able to spawn successfully. So another reason, I mentioned this earlier, is that they do support valuable recreational fisheries. In the Florida Keys Flats fishery, the annual economic impact is $465 million every year. That's bonefish, tarpon, and permit. And they're all catch and release. So in that sense, it's sustainable. In the Florida Everglades, which bonefish is a part of that fishery, it's $1 billion a year of economic impact, which is a big number, obviously. Statewide, saltwater recreational fishing has an annual economic impact of $7.6 billion a year. Now, you think about Florida's economy, you're starting to match some of things like the orange industry and others with recreational fishing. You go outside of Florida, it's also big. Belize, it's over $50 million US dollars a year, which is $100 million Belize dollars a year, which is huge for their economy. Bahamas, it's over $140 million a year. So without going through more data, you kind of get the point. As a catch and release fishery, these guys are supporting a lot of local economies in hopefully a sustainable fashion. Okay, now switching gears to talk about some issues we're starting to have with habitat. So first, some kind of generalities about habitat and fisheries. For most species of fish, the amount of habitat influences how big the population can be. So even if we never fished for them, never harvested them, a limiting factor in their abundance is going to be the amount of habitat. So for example, if you take away the juvenile habitat for bonefish and have low juvenile survival, you're never going to have as many adults as you should. The second generality is not just a habitat amount, but the quality of habitat. Or you can have healthy and unhealthy habitats. That's going to influence how quickly fish grow, how big they get, how they'll be able to avoid predators, and of course, then how many there are. And habitat connectivity is also important. Remember I showed you that picture of the habitat mosaic? If those habitats don't have some connectivity for the bonefish to actually kind of connect all those dots, then you could have all those habitats spread different places, but if they have to run the gauntlet to get from one habitat to the other, that's also not going to be successful. It's not just habitat, there's a lot of qualifiers here. So this is a problem, because in Florida, we have some serious problems with our fisheries based on habitat. And I'd say that we're running a deficit. This picture is from Fort Lauderdale area. It used to be a mangrove swamp. The dark green there is the mangroves and the golf courses, which of course a lot of water quality issues immediately adjacent. So Florida has already lost around 50% of its mangroves from pre-development times. We've lost over 9 million acres of wetlands. This may be top in the United States for acreage of wetland loss, or it's not. If it's not, it's in the top 5. We've already lost 2 million acres of seagrass and growing with some of the issues. Indian River lagoons, great example. In some places, up to 80% of the oyster reefs have been lost. Northern Indian River Lagoon is a good example of that, right? And these are all habitats that fish depend upon. We also have trouble with freshwater flow alterations. Everybody's heard about the Lake Okeechobee issues, right? But it's not just about the pollution that's in the water. It's about that if you change the timing, the amount, and the location of where fresh water flows into the estuary, you're going to change the estuary ecology, period. You could have the most pristine fresh water coming out of Lake Okeechobee or any of the other places coming in the Indian River Lagoon, and if that changes the salinity of the fresh water, the salinity regime of the water in the lagoon or the estuary, you're going to cause a lot of issues. For example, if you expose turtle grass to fresh water for more than a couple weeks, it's going to die. Same with oysters. And that has nothing to do with any of the pollutants that are in it. That's just purely changing the freshwater flows. And there's a lot of that going on in Florida. But we also have the water quality issues. A lot of nutrients, which everyone's heard about from various sources. Herbicides and pesticides are another big one, and I'll talk about this with bonefish in a moment. And heavy metals, which once they're in the system, they really persist, and they cause a lot of issues with reproduction and growth. So just the changes in fresh water are kind of that first impact. These water quality issues are more like the legacy. You get all those nutrients in the system, and they're going to stick around for a long time before they can actually cycle out. So they stick around even if you stop the issues. So all this has resulted in Florida of a significant bonefish decline in the Florida Keys. On the right are graphs that show the results of interviews with fishermen and fisheries surveys from the 70s until today in Biscayne Bay, Florida Bay, and the Florida Keys. And in every case, the top one is overall quality of the bonefish fishery. The second one is bonefish size. And the third one is how many bonefish they call shots on. It's entirely a sight fishery for bonefish. You're out on a flat skiff. When you see the fish, that's when you cast to them. So you rate the quality of your fishery by how many shots you get at bonefish, which basically means they're seeing fewer and fewer. So 3 different ways to characterize a fishery all show long-term declines in the fishery. A lot of that has to do with the habitat. One is water quality, and we're doing a lot of research on this. Folks at Florida International University are pulling together 25 years worth of data on water quality and seagrass habitat. There's been a lot of seagrass habitat changes. I think the number now is 60,000 acres of seagrass in Florida Bay is gone from the most recent die-off, which is due to water flow changes. A lot of habitat loss in Florida Bay, the Florida Keys. All you have to do is drive down there and see some of that. Contaminants, we're finding that copper is super abundant in a lot of the sediments in the Keys. A lot of that's from herbicides, copper sulfate, that they spray in the ponds to get rid of a lot of the algae. Copper sulfate, if you spray that directly on invertebrates, you'll kill them. Bonefish eat a lot of invertebrates. So we're looking at that as an issue as well. And then, of course, the connectivity I've already told you about between Cuba, Mexico, the Keys, et cetera. And we're having those conversations with those countries already. And finally, catch and release practices. Until recently, people didn't know that the way you handled fish affected whether or not they survived. So if you keep a bonefish out of the water to take pictures for 30 seconds or more, you increase the likelihood it's going to die by 6 times. So 600% increase in mortality of that fish just because of the handling. And the same is true for a lot of other fish, redfish, snook, tarpon, et cetera. So we're working on that to reverse that with education as well. So that brings us to what we're doing at Harbor Branch now, is with the population decline, some people say it's declined by 90% since the 1970s, we've got a serious issue. What we think is going on with the spawning is that you have to have a minimum number of bonefish in a spawning aggregation for it to be successful. If we watch a spawning aggregation that has, say, 5,000, 10,000 fish in it, we see very little predation. There's a lot of sharks around, a lot of barracuda, but they seem kind of overwhelmed by the vast numbers of fish spinning in circles. If that spawning school is only 300, 400, it's actually decimated by the sharks, the barracuda. You can watch it happen. And very few fish make it to survive, much less try to spawn that night. So our big problem with the Keys is not only do we want to restore things, but we want to restore the habitats. We want to restore the fish population so they can be self-sustaining. So we are working now with the state and others on water quality improvements. Bonefish Tarpon Trust has been pushing pretty hard on Everglades restoration policy. Habitat restoration and protection, we're working on a lot of that for juvenile habitats, especially for tarpon, not bonefish, but we're getting there. And different other ways to improve management. Part of that is the whole catch and release practices. But as I said, if you don't have enough fish spawning, you might not be able to sustain your local population if your local population is coming from local spawning. We think that at one point it probably was. Right now we're not so sure. The other thing about this type of spawning behavior is those migrations from the home range to the spawning site, we think that is a process called social learning. So let's say you're a newly mature bonefish, you're all excited, it's time for spawning, but you have no idea where to go or what to do. And then all of a sudden this female goes by leaking all kinds of hormones, you get right in her slipstream, go to the spawning spot, and that's imprinted. And that's how you know to go back there every year, whether that same female comes by or not. If the population gets so low that there aren't enough old fish to teach the new ones where to go, then you have another problem. And this has been documented for Nassau grouper, for example, in the Caribbean, where the Virgin Islands made Nassau grouper no harvest in 1985, and they still haven't come back. I've spent thousands of hours diving in the Virgin Islands. I think I've seen 2 Nassau grouper, and they have the same type of spawning behavior as bonefish. So to fix all, to address that aspect of restoration, stocking might be necessary. And we consider it a tool in the overall restoration toolbox. It's not an approach to say stocking fixes everything. If we don't fix the habitats, we don't fix the causes of the population decline, stocking's not going to do much for us. So it's one piece of a pretty complex puzzle. It's also for a finite time period. So what I'm going to do now is going to go through what we're doing here right now at Harbor Branch. This is really the only geeky slide I have. I apologize, but it's the best way for me to go through it. This is a flow chart of what we're all supposed to be doing. We had another meeting today to talk about what we're all supposed to be doing. And it's a challenge. So, and I'll go through some examples of this, but basically we're looking at what hormone treatments we can use for bonefish to induce them to spawn in both the lab and in the field, trying to figure out what type of physiological chemistry they have going on to get them to spawn. How do the eggs develop? How do the larvae develop? What do they eat? All those types of things. It's a pretty complex and challenging process. And I should also say that no one else in the world has done this. So we're going to take on the challenge and get it done in the next 4 years. So one of the things that we're doing is examining wild bonefish in association with spawning aggregations to tell us what to do in the lab. So we don't know, for example, what the hormone structure is in bonefish, right? When they're non-spawning form in July, the hormone levels are probably different than when they're getting ready to spawn in October or November. So we are looking at using blood tests, taking out, withdrawing blood, analyzing that blood to see what hormones are being released during spawning, getting ready for spawning. We're also cannulating eggs. You don't have to kill fish to get eggs anymore. We can actually extract them using a syringe and tube. I'll show you a picture of that in a second. In the lower right here are some tags, some geo tags that we're going to use to try and get some information on their behaviors. All of this is to inform what we're doing in the lab so we know whether we're doing the right thing. If we can actually get the fish in the lab to do what they're doing in the wild, then we're in good shape. And as I said, the depth that they go to provides a real challenge. Trying to mimic that in the lab may be very tough. Getting the amount of atmospheric pressure at 200 feet is going to be tough to do in the lab, so we have to figure out ways around that. But that may be something we have to mimic. We're also looking at what it is that the bonefish put into the energy for eggs and spawning. The picture here is eggs that have been extracted from the bonefish by cannulation. They're being put into a vial to be analyzed. And what we're trying to figure out is what are the energetic requirements, which the energy is stored in lipids and fatty acids that are used in the eggs to make the bonefish ready for spawning and make the high quality eggs. Right? There's something that we call a condition factor in fish, and that's essentially the ratio of length to weight. So if you have a long, skinny fish, it probably doesn't have a lot of fat reserves to help to build a lot of quality eggs. If you have a chunkier fish with fat reserves, it's going to be a lot better spawner. We have to figure out exactly what those combinations are. So we're undergoing now, getting started on lipid and fatty acid composition. What type of these lipids and fatty acids are in the eggs, and how do they change over time? Compare them to the wild females again, and how are they looking in the lab? Because a lot of times you put fish in the lab and they get stressed out and they won't behave like they're supposed to. I wouldn't either. And then look at how they change over time within the spawning season. So this is well, I guess I would lie. This is a second geeky slide. Essentially, each color is a different type of lipid or fatty acid. That's all you have to get out of it. These are 3 different locations on Grand Bahama Island from this past year. And you can see that the composition, the relative proportions of the different fatty acids differs just from 3 different locations. And these were sampled over a period of about a month. So there's a lot going on that we need to understand. If there's this much variation in the wild population, this gives us a cue that these guys are pretty complex. They're not going to be as simple as some of the other stuff like redfish. It's going to be a lot more complex. We're also doing a lot of work in the field. These are 2 bonefish that are in an inflatable pool. This is in the Bahamas. In the background is a pre-spawning aggregation site. So these are actually collected. They're ready to spawn. They're at the site. They're ready to go. So what John is doing there in that photo is getting some hormones ready to inject with hormones to see if he can induce the eggs into reproductive state. So we're looking at the behaviors, the specific time periods about all the different behaviors. Can we get them to spawn in captivity even in this type of situation when they don't have 10,000 other fish around them? And if we can it be induced either by hormone injections or by environmental controls? And I'll get to the environmental controls in a moment. So this is a slide from the same fish of pre-injection eggs on the left, and on the right is post-injection. And there's some development that's been happening. A lot of oil globules are forming, which is moving towards being ready to spawn. The problem is this is as far as it got with this fish with this hormone injection. So we induced something, but not enough. But considering that we're just getting started, that's a pretty good start. And we're trying a lot of different hormone treatments to see what works best. Given the fact that bonefish are so old and are so different from any other fish, it's not something we can take what we know from other species and just plunk it onto bonefish. We're also taking what we're doing in the field and we're emulating that in the lab. This is here at Harbor Branch doing the same thing, withdrawing eggs by cannulation, doing some hormone injections, monitoring the fish over time to see if we can get them to spawn in captivity. We're also doing a lot of other non-invasive techniques. Sonogram, for example. On the left is a male, and on the right is a female. So we can see developmental stage and whether the male or female without the old school way of essentially slicing them open, which doesn't help with the whole spawning deal. So we're getting a lot better at this kind of non-invasive stuff. For environmental controls, we've got a number of different tank systems where we can control the length of day, the intensity of light, water temperature. We can even do a lunar phase, so full moon, new moon, laying all that over. And one of the tricks that's used for other species is if you speed up summer, since these guys are winter spawners, make summer happen really fast and then try and get them into their winter spawning mode, maybe you can kind of induce that at a more rapid pace than you might see in nature. So there's a lot of science madness going on back in those greenhouses in the back of the property. We're also looking at early life stages. That larvae I showed you, no one really knows exactly what's going on, what it eats, those types of things. What are the environmental conditions for the eggs to be successful after they come out of the female even? Is there a temperature or salinity? And the same thing for the larvae. So the questions, among the questions we're asking is, what are the effects of different light levels, day length being one, water temperature, salinity, how do they affect the eggs, how do they affect the larvae? For other fish, it's been shown that temperature differences can change their growth rate significantly, which then affects their survival once they become juveniles. And finally, how long does it take them to develop? Remember I said that the larval stage is 40 to 70 days. That's a pretty wide range. So can we shorten that, get that all the way to the short scale so we can get them out of that phase, which is harder to feed, and into the juvenile phase where it's easier to feed? Those types of manipulations are the types of things that we're starting to address. And the big question is, what do larvae eat? It's pretty amazing, right? Here we are in 2017, and we still don't know what these guys eat. But there are some ideas. So this is a close-up of a bonefish larvae. One theory is that they eat what's called marine snow. And that's in the upper left here. And marine snow is basically aggregates of bacteria and mucus and slime and fungi and all kinds of things that just kind of coagulates into these white blobs that if you are diving in the open ocean, deep blue, it does look like snow just slowly falling from the surface towards the bottom. And the theory is that these larvae and others similar to them just kind of glob up to these snowballs, snowflakes, and just kind of churn away at all that mess that's in those aggregates. On the lower right is a slide, not a bonefish larvae, an eel larvae, that was fed squid paste with red dye in it so they could actually see if it was ingesting it. And it did. So there's some work that's being done that we need to build upon. Now we're kind of moving along through the life cycle, juvenile growth and nutrition. The lower left picture you've seen before when they first come in. On the lower right, we have bonefish right here, open sandy bottom. They mix a lot with mottled mohara. And from the top, they look very similar. The theory is that if they get in those schools of mohara, predators can't find them as well. As they grow older, the bonefish get in their own schools and set the mohara aside. So open sandy bottoms, we know. We also know they mix with mohara. And we know that they eat a lot of amphipods. These little guys live on and in those sandy sediments. But the interesting thing is that the amphipods they eat are different than the species that the mohara eat. So they're not competing directly for food, which means that the bonefish may be very specific on which types of amphipods that they eat, which would be very frustrating. But hopefully we can convince them to eat our amphipods once we get them into the tanks. And then we have the challenge of as they grow, their diet's going to change. Remember I showed you 16 inches over and under? There's a lot of diet changes going on before that too. So we can't just keep feeding them amphipods. As they grow older, we have to figure out other things that they can eat to maximize their growth, maximize their health, maximize their condition factor. So finally, getting into the stocking approach. This is your conventional stocking approach, right? Trout truck up in Michigan, getting ready to dump all these trout. That's not what we're going to do. We're not into the stock enhancement, put and take fishery, where people follow around the stocking truck. It's a completely different approach. First thing we have to do is in the Keys, and this process is ongoing now, is identify the specific habitats and locations where juvenile bonefish, the size you see in this picture, are found. These guys are probably just a couple months old. Where are they found? Identify those locations, and that's where we stock. We also look in those locations, what kind of prey are in those areas, and is that prey that bonefish will be able to eat and grow with. With so few bonefish in the Keys, we can't just go there and find juveniles and say, hey, this is what they're eating, and this is where we think a lot of them are. There's not enough there. It took us 2 years to find 6 juvenile bonefish. So we're kind of redoubling those efforts. We also have to figure out if certain areas have more predators than others. And if they do, we avoid the high predator locations. And see what other species are there. If mohara are there, then we're in good shape. If for some reason mohara are not there, then it's probably not good for the bonefish as well. So much more specific than your typical stocking effort. We also have to figure out what size to grow them to before we release them so they can avoid predation and make it on their own. And believe it or not, we'll also have to train them. A lot of research has been done, for example, on snook. If they just dump juvenile snook into mangrove creeks, which is their juvenile habitat, and just let them go, most of them get eaten within the first couple of days. They just don't know what's going on. But when they've been trained, in essence, they put them in large net enclosures in these creeks for 3 to 4 days. They kind of learn that this is not the tank anymore. They have to find their own food. And then they also see predators kind of lurking around the outside of the net. And so their survival goes way up. So we'll have to do that with bonefish as well. Much more complex than your typical thing. So this is an image of Sugarloaf Sound in the Florida Keys. One of the places that we'll probably start working here pretty soon. This is one of the places we have caught juvenile bonefish, but it's a big area. You can see some of the channels, the darker lines there are channels that would deliver larvae, so that's a good spot, good sign. But where in here would you want to put juvenile bonefish is the big question. Some of these areas you can't tell from here. In the lower portion, this area has a lot of upside-down jellyfish, Cassiopeia jellyfish. And they sting when they get on you a lot. But we've also found that if you stir them up, the nematocysts, the stinging cells that come out of those Cassiopeia, will cause bonefish mortality even of adults. So probably not the best place to put juveniles. So a lot of that type of work is getting started. So basically in the end, and this is a picture of a pre-spawning school of bonefish swimming around an acoustic receiver there at the bottom. It's about getting the population in the Keys right now back to this level, but also knowing that there's a lot of other fisheries and habitat loss issues going on in places like Belize, where the government, unfortunately, is selling off a lot of their coastal submerged lands to corporations like cruise ship companies, which is now being dredged, et cetera, to make fancy cruise ship resorts. And that is impacting the habitats. So as we learn this in the Keys, it's essential for us to do this so that we can restore the Keys population. But this is also going to be very applicable throughout the Caribbean, if not in other places in the Pacific, where they've had some pretty significant declines as well. So with that, I'll end. This is a collaboration with Bonefish Tarpon Trust, Harbor Branch, and the National Fish and Wildlife Foundation, which saw the importance of this study and is providing a good bit of the funding. So thank you.